It is estimated that the average human being is composed of 30 trillion cells, but every organism, including you, started off as a single cell. That single cell had to grow and make copies of itself in a process known as the cell cycle. There are two stages of the cell cycle, interphase and mphase. Most of a cell's life cycle is actually spent in interphase, where they are growing and synthesizing DNA to prepare itself to divide in mphase. During gap 1 or G1 of interphase, the cell carries out normal activities and functions. The cell will start to increase in size, and the number of organelles also increase. During S phase or synthesis, the cell will make copies of its nuclear DNA. By the end of S phase, the cell will have 2 complete sets of DNA, and it is just about ready for cell division. During gap 2 or G2 of interphase, the cell will carry out normal functions, and additional growth occurs. The cell will undergo a critical checkpoint before it enters cell division. It has to make sure that its DNA has been undamaged and replication has gone smoothly. Each step of the cell cycle has quality control checkpoints carried out by proteins known as cyclins, which make sure the DNA has been replicating normally and that the DNA is undamaged before continuing on to the next phase. M phase is the process of cell division, and there are 2 stages. There is mitosis, which is the division of the cell nucleus and its contents, and there is cytokinesis, which is the division of the cell cytoplasm to create 2 identical daughter cells. Although mitosis is a continuous process, there are 4 stages, which you can memorize with the acronym PMAT: prophase, metaphase, anaphase, and telophase. The DNA in your cells are loose strands, and they would stretch out to over 3 meters or 10 feet. As a result, during the first stage of mitosis, known as prophase, the DNA will condense itself by wrapping it around proteins known as histones. It forms tight coils known as chromosomes in this process. Furthermore, the nuclear envelope starts to break down. Centrioles then start to migrate to opposite poles, where they form spindle fibers which are made of protein that eventually pull apart the chromosomes during the later stages of mitosis. In the second stage of mitosis, called metaphase, the chromosomes start to align themselves along the cell's equator, or along the middle. Spindle fibers then attach to the centromeres and begin pulling the chromosomes apart. In stage 3 of mitosis, called anaphase, the sister chromatids continue to be pulled apart by the spindle fibers, and they separate to opposite sides of the cell. The cleavage furrow also forms, which is the beginning of how the cell begins to divide and split in half. In the 4th and final stage of mitosis, called telophase, you can see the nuclear membrane start to reform, the spindle fibers fall apart, and the chromosomes start to uncoil as the daughter cells begin to take their own shape. Once mitosis has come to a completion, cytokinesis then divides the daughter cells into 2 parts. In animal cells, a cleavage furrow forms, but in plant cells, a cell plate forms along the equator, separating the cells into 2. Let's go ahead and summarize this with an animation of mitosis. And remember, in mitosis, there are 4 stages: prophase, metaphase, anaphase, telophase. And remember, the acronym is PMAT. So during prophase, you have your sister chromatids, which is the duplicated portion of your chromosomes. And you get these things called spindle fibers that start to form from your centrioles. Okay, the centrioles are a really important organelle in cell division. So the centrioles will then migrate to opposite ends of the cell. And now these spindles start to come out. They're made of microtubule filaments, and they start to attach to the kinetochores, which are found on the chromosomes. Okay, and then it starts to line up along the center or the equator during metaphase. And now these sister chromatids get pulled apart by the spindle fibers during anaphase. So anaphase, you'll start to see them migrate to opposite ends of the cell. And telophase, you'll see the cleavage furrow start to form, and something else called a contractile ring, which is made of proteins, and this thing eventually severs the 2 daughter cells, so then they can each live their own life. Okay, and so this is nearly the end of mitosis. Okay, you've duplicated your DNA and you split them up. So now each cell has their own set of DNA. And in cytokinesis, you finally see the complete separation of the two cells. So there you have it. But in plant cells, something called a cell plate forms because they can't form that contractile ring. So a cell plate will then divide these two cells and they separate. To give you a short summary, just understand that there are two stages in a cell's life cycle. You have interphase and you have M-phase. A cell spends most of its life cycle in interphase, where it spends its time growing and duplicating DNA so that it's ready for mitosis. You can think of mitosis as a well-choreographed event of cell division where each daughter cell receives its own complete set of DNA. Finally, you have checkpoints which are carried out by certain proteins and enzymes known as cyclin and kinase, which make sure that the DNA is being replicated without damage during each step of the cell cycle. Thanks for tuning in and watching my lesson on the cell cycle and mitosis. I'll see you next time on Win Biology.